reason my computer decides to hang the minute I want to switch on my video. So I think that's a reason why. But thank you for joining us at our Drug Discovery Learning Series brought to you by SG Innovate. Just quick housekeeping rules. Please note that this webinar will be recorded and all participants except for the presenter will be on mute. Lastly, should you have any questions during the webinar that you would like to ask Ashley, please feel free to post it on the Q&A function at the bottom of your screen, and we will be more than happy to pick up most of the questions by the end of this seminar. As some of you may be aware, the SG Innovate has recently launched our Tech Talent Central initiative, and together with it, we have also announced our new talent program, the Helix Immersion Program. If you have not seen us sliding through the slides in the last few minutes, please do come and visit us at our website at SG Innovate and just search, Helix Immersion Program. You'll be able to find more of our program. This whole program is about bridging the biotech talent for Singapore and building new talent needs for our ecosystem. Today, I'm extremely excited to have Mr. Ashley Burns, the Chief Scientific Officer of Exo Biosciences. Exo Biosciences is a company that harnesses iPS cells technology to produce better human models and help to accelerate drug development with the next generation in vitro disease model. Ashley has over 30 years of experience in drug discovery, and in the last 7 years, he has been spending a lot of time using iPS cells as a different alternative to disease modeling. Prior to this, Ashley was at the GlaxoSmithKline for more than 20 years, where he led project teams across a range of diseases, including respiratory and neurosciences. We are very honored to have Ashley here today sharing his talk on better human models using iPS cells, what we have learned, where are we, and the future. Ashley, the floor is yours. Take it away.
Thank you, Vanessa. Hello, everyone. Thank you for coming to hear me talk today. I've got a series of slides that I shall be going through. The data that I will be showing, the assay data, has been generated mostly by the team at Axel, and I'd like to thank them for their efforts in providing me with that data. There are one or two other bits and pieces from other groups that I will point out where they've contributed to that. So I'm going to walk through a series of different opportunities and uses of iPSCs, but I'm going to start with very much a brief introduction around why iPSCs, what the value they can add over and above current methodologies. As Vanessa said, I've been in this space for quite a long time. I've been working in drug discovery for quite a long time, and obviously there's always been a gap in what we're trying to do with regards to drug discovery and failure of clinical molecules when they get to the clinic. The failure rate is quite high. So what we're talking about here very much is how do we get from what we do on a daily basis in the lab with our pipettes and our cell culture methodologies and our assays to get to clinic and basically help patients. So where we stand at the moment, and this number hasn't really changed much over the last 20 years that I've been working on in the drug discovery space, and you can see from these numbers that we still see a huge number of drug candidate failures, and a significant number of those failures are down to lack of efficacy in the clinic. And what we mean by that is the molecule where it was intended to target and give a response doesn't give a response. On top of that, there is a whole series of compounds which also have a particular problem with regards to cardiotoxicity, and this has been an area of focus very much for the FDA in the US but also in Japan and other areas. And I'll talk very briefly about how iPSCs can be used to help in that space too. So why iPSCs? So here's a quote around where we currently are using animal models and what's the limit of animal models in this in the drug discovery paradigm. And as it says here, mice actually don't get Alzheimer's. You can modify them so they can get Alzheimer's by massively overexpressing proteins. But actually, a normal mouse won't get Alzheimer's, nor will they get asthma. They won't get a whole series of diseases that humans get. And even when people have used and do use non-human primates in animal models, they aren't always a great translation to humans. So we need better ways of doing drug discovery, and the identification of iPSCs and the use of iPSCs, we believe can help drive that. So coming on to iPSCs then, and how do we use iPSCs in drug discovery? So I think just a brief background around what iPSCs are and how they can be used. So iPSCs themselves were invented or defined by Shinji Yamanaka and his team in 2006. And you can see the seminal paper from that group on the left-hand side. And then on the right-hand side, there's I've also then highlighted CRISPR now as a gene editing technology. Now, the value of CRISPR and iPSCs together, I think, is an important consideration. I think the other comment I would make is that neither of these two technologies are start from a zero baseline. And what I mean by that is stem cells and their use of stem cells has been around long before Yamanaka identified ways of reprogramming human tissue. So human ES lines preceded iPSC lines, and mouse ES lines have been used for many years. And so, you know, these developments are very much building on previous understandings and knowledges, but these are a huge step over and above what was previously used. CRISPR is the same sort of thing. So gene editing and methods of gene editing have been predate CRISPR by quite a few years. So homologous recombination in mouse models has been used for many years to knock genes out. And things such as zinc finger nucleases have been around for at least 10 years before CRISPR was identified. But what these two technologies do is they springboard you from methodologies that are not really that usable. You can use them on the odd occasion, but they are highly expensive, to being far more usable in the lab on a more regular basis. So just to highlight those two technologies. So how do we currently use iPSCs in the drug development journey? So this is the standard chevron diagram that you may see on regular basis, and is what we use and how we understand the drug development process. So from the left to the right, we are using iPSCs to identify targets and understand biology and disease. They are currently being used along with CRISPR to knock genes out and understand how genes themselves, specific genes themselves, interact in particular diseases. We at Axol are focused on neuroscience and neuroinflammation, and also motor neuron disease, and I will talk a bit about those two diseases and some of the models we use in that space. Then they are currently used and are used in big pharma for compound understanding and triaging compounds, all the way from to SAR and then up to candidate selection. Now, once you get to candidate selection, and what we mean by that is you've gone from your screening library, which could be 2 million compounds, and you've got down to your one compound, then what you see is generally a drop-off in the use of iPSCs as it stands at the moment. And certainly where we see, and what I'll talk about with regards to the future, is how can you help in identifying and reducing that, that attrition that we talked about at the start of this presentation. And how they may be able to be used post-trial to understand failure rates as well. So I briefly talked about animal models and how they are, they have their limitations. I think one of the things that's happened very, very recently is this particular bill that was passed in the US, which is known as the FDA Modernization Act 2.0, and how this changes the paradigm for where we currently are, or where we currently have been, is this basically enables drug discovery to happen without the use of animal models now, or even animals in the development phase of the work. Now, how it doesn't mean that animals are not going to be used, but it enables drug companies to think about how do we use in vitro models and better in vitro models to de-risk drug discovery and de-risk clinical trials. So this is actually, you know, this is a big step, and people in our space have been calling for this for quite a long time. But this really is an inflection point in the drug discovery process. So I'm going to now move on to iPSCs themselves and talk a little bit about them, what they are, what some of the challenges are, and why we're focused very much on trying to build a quality cell line and quality reagents in order to enable this type of work. So generation of iPSCs, they are not the easiest thing to generate. If those of you that may have worked with hex and CHOs and can passage those relatively straightforwardly, iPSCs are far more technically challenging. However, they do offer this great opportunity for drug discovery. So the QC of iPSCs can be challenging, and you need to keep an eye on that. Keeping on top of the passaging methodology and the details around controlling spontaneous differentiation, and I've got some slides on that. Optimization, the maturation time. Some of the methodologies that we use for the maturation and the differentiation of these cells are really, really long in culture, in terms of months in culture in some cases, and certainly 2 or 3 weeks. And that can be a challenge, certainly, for maintaining sterility. And then I'm going to talk about some of the cellular assays that we use to validate the iPSCs that we differentiate. But iPSCs are not just about the cells themselves. There is a whole piece around the media and the matrix that you use to stick the iPSCs down onto culture dishes, which takes quite a lot of time to validate and qualify. Some of which is often overlooked, but is actually quite important, and I will briefly talk about that too. So I mentioned iPSC quality control, and what do we do around, how do we generate an iPSC line? So I'm not going to talk about the ethics and obtaining iPSC lines here, but what I've shown here is a C of A, so a certificate of analysis that we carry out on any iPSC line that we generate for a client or for ourselves. And you can see this is just one page of about 4 pages, but this is the summary page. And what you can see on the left-hand side here is all the different tests that we are carrying out on the iPSC in order to make sure they are as we expect them to be, which is pluripotent. And what I mean by that is they will go to the 3 different types of lineage, so endoderm, mesoderm, and ectoderm. And you can see on this particular case, we had a bit of an issue with the ectoderm readout, so that assay was actually had to be repeated. But here you can see all the numbers of different tests that we carry out and try and keep on top of. Now, one of the areas that we are focused on is very much trying to maintain the genetic normal cell number and chromosome number in the cells. Cells generally are always trying to find a way to grow better and grow easier. And on the right-hand side here, I show an example of, this is an iPSC line. It's not the iPSC line from that C of A on the left-hand side, but this is data taken from an iPSC line where we have QC'd the karyoology of this line. And what you can see is an expansion and a trisomy in chromosome 13. Now, there are a number of different mutations that can go on iPSC lines. Chromosome 20 is another standard expansion that you can see. But if you keep passaging these cell lines hard and splitting them hard, you will end up with this type of mutation, because the cells basically try and find a way of surviving. And that's what you see here. So maintaining QC and on top of the QC is really, really important when you're using iPSCs. So that's the QC part. This is just growing the cells. So what I'm showing here is this is the same line. So this is exactly the same iPSC line, grown in slightly two different ways and two different methodologies. So on the left-hand side, we have a method where we have been splitting the cells quite harshly and single cell passaging these cells. And on the right-hand side, we have a picture here of cells that have been split far less harshly, and they are called clump passage. So the cells aren't dissociated to single cells, they're left in clumps. What you see on the left-hand side is the cells spontaneously differentiating, and they will do that because, again, it comes back to them trying to survive and trying to maintain themselves. And that makes it really, really difficult to take these cells through a variety of different differentiations. So the cells on the left-hand side, we would not use in assays and differentiations, and on the cells on the right, we would. So this is quite a difficult thing to keep on top of, and does rely on quite a high level of expertise. It's certainly a step above working with HEK cell lines and CHO cell lines and standard cell lines that people have been using in the lab for many years. So that's the iPSCs. When we come to talk about differentiation and taking these cells to different lineages, to neurons, to astrocytes, and to microglia, which we'll talk about later, the media themselves and the reagents we use to do that can have a big effect on what you see. Now, this slide here shows two different suppliers of VEGF, which is a reagent that we use in this particular differentiation. So this is taking these cells to a microglia diff. And on the top, so these are allegedly exactly the same reagent. On the top, you can see that the cells have balled up and have started to slowly, much more slowly differentiate. And in this case, that's not what you want to see. What you want to see is the structures you see on the bottom, very much what we call the fried egg structure. And what you see is this is done in exactly the same time in parallel and driving these cells to this particular hemangioblast morphology, which are then generating monocytes from this particular structure. Now, the differentiations here are exactly the same. The concentrations of the reagents we use exactly the same. But what it does highlight is that the differentiation itself, just having the ability and understanding the differentiation and being able to do it is not good enough. You actually need to understand different supplier reagents and how do they affect the differentiation. So these are the sort of things that we look at on a regular basis, and I would suggest it's the sort of thing that if you're doing any work on iPSCs, look at more than one supplier of reagents, because your differentiation may well fail, not because you're doing it wrong, but because something you've brought in isn't quite behaving as you want it to. So that's differentiation and trying to drive to better differentiation. Okay, so this is this slide I put up here is very much around trying to make use of iPSCs easier. And I talked about the length of time it takes to differentiate cells and using these cells. So what we are trying to do, and as a company we try to do, but it's what I would say the whole of the industry and using iPSCs are trying to do, is trying to make iPSCs use easier. Now, if you're using differentiated cells and you've got to differentiate these cells for 56 days in culture, in a flask, or in a plate, that's quite a challenge. And so what we have done and what we're trying to do is to decrease that time frame. So this is motor neurons. These are motor neurons, and I've got some data later on motor neurons, but this is motor neurons. And a year ago, we were having to generate a 56-day protocol to enable us to get to a mature motor neuron. So this is a picture of HB9 staining, which is a particular marker of motor neurons. And you can see that they're all lit up. And although I haven't got all the data here, you have to take my word for it, but we have been able to get this to move this to an 8-day protocol and can see firing, and I've got some data on that later. But what we've done here is modified the media and spent quite a lot of time in trying to understand the media components that allow us to drive the differentiation. So in this case, we've changed the media components, and what we've been able to do is reduce that maturation from that 56 days to 8 days. So what I hope I've just sort of given you a brief highlight of is the effort and the work that we certainly put in, and I would recommend if you are using iPSCs and want to work with iPSCs, that you keep on top of a number of these very important factors, which can make a big difference in the assays that you generate, and also the physiological relevance of those assays. So that carrier typing data, for example, and keeping on top of the chromosome number, that can have a big effect on the expression profile of certain proteins. So now I'm just going to walk through a number of slides on physiologically relevant assays. Some of them I'll go over very quickly and pass over, and some of them I'll spend a bit more time on. So I'm just going to start off with a microglia differentiation. So I talked a little bit about CRISPR gene editing. The data I'm showing here is some work that we've done looking at gene edited cell lines. So in this case, what we've done is we've taken an iPSC line and we've taken a number of different gene edits from those lines. So in this case, we've got CD33, in which which the exon 2 has been deleted, and we've got TREM2, which is a particular marker and a particular protein in microglia, which has a mutation in. TREM2 is associated with frontal temporal dementia diseases, and CD33 has been identified as a protein which is actually protective against Alzheimer's disease. So if you look at these particular mutations, these are mutations that have been identified in patients, and in this case have been modified on a single iPSC line. And then you carry out a series of assays here. So what we've looked at here is release of IL-6 from these differentiated cells. And then we've also gone to look at phagocytosis of A beta 1 to 42. So that's amyloid beta, which is very much associated with Alzheimer's disease. And what you can see here is a significant change in a specific mutation in TREM2 with regards to its ability to phagocytose versus the other clones here. So what we're doing here is very much focused on differentiating these cells, taking them to a microglia, which are the immune cells in the brain, and looking at how they behave in isolated assays. So there's an example of the type of assays that people run and that we run on a regular basis to carry out drug discovery. So that's microglia. Here's another example of looking at disease modifications associated with motor neurons, and I talked about motor neurons previously. So motor neurons are very much associated with with ALS and motor neuron disease. Loss of motor neurons and motor neuron function is associated with the progression of that disease, and always leads to death in these patients. What we have here is a couple of examples of motor neurons on a electrophysiological measurement platform. So this is a multi-electrode array platform from Axion, and this is data we've generated in-house. What we're looking at here is two different iPSC lines from two different patients. So the person on the left-hand side doesn't have motor neuron disease, and the person on the right-hand side does have motor neuron disease. So these were identified by a clinician. We've taken those samples. We've then generated from the patient fibroblasts iPSC lines. We've then taken those lines and differentiated to motor neurons, and then measured the electrical activity of those motor neurons. And what you can see here is a significant difference in the behavior of those two different motor neuron material. So the material on the right-hand side, you can see very high spontaneous firing, not clustered and not controlled at all. Whereas on the left-hand side, it's much more controlled and far more regular in its pattern. So this is data that we've generated. There is literature out there. This is a C9orf72 mutation that this patient had. There is data out there on other ALS mutations, which looks very similar to this. So superoxide dismutase mutations and TDP43 mutations look very similar to this type of data. So that gives you an example of the type of work we can do on neurons. Again, disease relevant. And now I'm just going to very briefly talk about this one. I'm not going to go into any detail. There are QR codes on some of these slides, which will allow you to look at these in more detail if you want to. So this is coming back to my comment around using iPSCs and differentiated iPSCs in development and safety toxicology. So in here, what has been taken, and this is work that we provided ourselves to a company called Clyde Bioscience. They took the CIPA 28 compounds. So these are the FDA compounds which have been identified as compounds which have caused cardiotoxicity in the past, and are used as effectively a training set on cells to identify whether your cells meet the criteria or not. I'm not going to go into this to any great detail, but the bottom line is, if you look at the activity of these compounds with regards to their hERG blockade and their field potential duration, and effectively the QT, long QT signaling that you see of these cells, and you can identify the trouble and the issue you can get with cardiotoxicity using cardiomyocytes. So these are iPSC derived cardiomyocytes, and then tested with a series of 28 compounds, allowing you to identify toxicology issues with these compounds. This assay has been used across the board in big pharma, and is used on a regular basis to de-risk the compounds that they're identifying for the areas that they're working in, disease areas they're working in. So this crosses all disease areas. It's not just around cardiotox, it's in every single other disease area. So I've talked about all the reagents and the assays I've talked about so far has been single cell. So you're taking a motor neuron, taking a cardiomyocyte, taking a microglia and doing assays on it. But the real value in iPSCs, or certainly the direction of travel in iPSCs, is can you put different cells together, and how do you generate better models? So here is a series of different publications that exist, just some, but there are plenty out there, using different co-culture models and driving different assays, putting them together. So astrocytes and microglia. So I'm just going to highlight a couple examples of these. I'm not going to go into any great detail of these. The first one I'm going to talk about here is motor neurons again, and skeletal muscle. So this is a methodology where you put, this is a microfluidic device. So the picture on the left-hand side of the screen shows a microfluidic device. And what you do is you put your cells in those holes, or black holes, and they move into the channels, and then they cross those channels. So the channel on the green channel, in this case, is that we use for the skeletal muscle, and the blue channel is what we use for motor neurons. What you can see here in this picture is the skeletal muscle on the left-hand side. So this is the big picture I'm talking about now on the left-hand side. They're skeletal muscles, and you can see that the motor neurons have moved into the skeletal muscle and formed a synaptic motor neuron synaptic interface. And we can see that and stain that by looking at the staining at the bottom. So underneath the big picture is 5 different pictures. This basically identifies that what we have seen here is generation of the motor neuron end plate. We're looking at the staining of alpha bungarotoxin, which is a marker for synaptic marker. We've got NeuroN, which is the yellow one, which highlights that those are neurons. And then we have titin, which shows that the red are skeletal muscles. You put those all together and you can see that they all overlap. So what we're building here is a model for working on motor neuron end plate, which is very much what disappears in ALS, and allows you to look at your compounds working on the motor neuron end plate specifically. And you can take different disease models into this. So you could take the C9orf mutation into this model, which we've done, and you can take the SOD1 and TDP43 model into this as well. So now I'm just going to briefly talk about another co-culture model. So what you can't see here is the cells, but what I am showing here is the action of the cells and the assay itself. So on the left-hand side, we have cells which have been transvected with a lentivirus containing a calcium-sensitive fluorescent protein. In this culture are cortical neurons and astrocytes. And what you're seeing here is over a period of days, you can see on the left-hand side, you're getting the cells behaving and flashing and twinkling themselves, but they haven't formed any form of network together. And then as you move across that picture, you can see they've networked together and you get spontaneous firing all at the same time. And that increases over time, over the number of days. So when you look at that and you do data analysis on that, what you're seeing here is that firing event going on. And what you can see is each individual one of those colors is a specific cell and a firing event going on for each individual cell. So you can see at the top it says active object is 375. So in this case, the INCUSI has identified 375 individual active firing events. And the one in the middle, it's identified 763, and then there's 594 on the one on the right-hand side. What I'm showing here is an example of if you put 2 cells together, what you're able to do and what you can drive. So what we're looking at here is that burst firing pattern and how that affects duration and function. So how do we use this in models and use this going forward? So what we have done, and again, I haven't got the time to show the data here, but what we've done is we've actually put multiple cell types into this. So if you put cortical neurons and you put inhibitory interneurons and you put astrocytes, you see a similar firing pattern, but a much reduced signal. If you then add astrocytes, microglia into this process, what you see is a massively reduced signal and a change in the firing pattern of these neurons. So what that allows you to do is, if you have different disease models and different behaviors of different neurons. So for example, if you take a frontal temporal dementia neuron, which you see a decrease in the firing rate and a loss of that firing, and you put a microglia on that, you can see a significant change in the behavior of those neurons. Okay, so I've gone through quite rapidly a whole series of different assays, just to really highlight the type of work that we're doing and the type of work that others are doing in this space, and how we can use these models very much to replace animal model usage, and very much identify better ways of generating drugs for clinical trial work. Okay, so there's very much a highlight picture of iPSCs, the assays that are run and that people run and trying to drive this forward. You know, the title of the talk very much had look into the future associated with it. So how do we see this playing out as we move forward over the next 5 years? So I talk very much around how we can look at different individuals, and we take different patient samples and moving this forward, and showing that each individual patient can show a phenotype. So if you then take a more holistic view of how that looks, if you were to take hundreds of iPSC lines from a patient cohort, differentiate those and run assays on them, you could enable a number of different things. One of which was you can de-risk a clinical trial. So Alzheimer's disease has a litany of clinical trial failures behind it, mostly because nobody really understands what causes Alzheimer's disease. And a lot of it is driven by sporadic events. It's not driven by a specific gene. So the motor neuron example I gave was very much a gene-driven event, but in motor neuron disease, only around 5% of the whole of the population that get motor neuron disease have a genetic association with it. A lot more are environmental. I'm sure some of you are aware of a number of sports people, particularly rugby, American football, and soccer or football. All have people of well-known sports people that have ended up with levels of motor neuron disease and diseases associated with neurodegeneration. And they aren't generated by a genetic mutation per se, they're generated by the environmental factors. So to be able to try and identify what's driving that is really quite difficult. So can we de-risk a clinical trial by taking 100 lines from motor neuron disease patients and carrying out the type of assays I just talked about and putting a compound on and saying, okay, I see this only working in 10% of the population of my 100 patients, or I see it working in 50% of the population of my patients. What does that allow us to do? It allows us to enable better clinical trial design. And so the problem with those numbers are, it doesn't mean that there isn't a signal in there, but the signal is so small that it isn't big enough to drive an event which is statistically significant. So certainly where we see this space in the drug discovery space is very much trying to drive to what we call clinical trial in a dish, allowing drug companies to de-risk a clinical trial before they even get to the point of doing a clinical trial, because clinical trials are tens of millions of pounds, and the cost of failure is really quite high. So that's clinical trial in a dish. I think a couple of things that I would just like to highlight and very much advocate. So if you're doing clinical trials in dish, what you need to be able to do is very much get a good representation of the population. I've talked about Alzheimer's disease and the failure rate in Alzheimer's disease. One of the biggest problems that we have in some of these trials is that there are some groups that are underrepresented in clinical trials. And I've just got a couple of slides on that, one of which is women in the female population. So there are sex differences between between male and female in different AD forms, and this is an example of a paper that calls that out. And I'm very much calling out effectively for more people to put themselves forward for clinical trials, but in my space, therefore providing samples to allow us to generate iPSCs. Representation of females coming forward to donate samples for iPSC work is much less than it is in the male population. And that's not just in the UK, that's also in the US, and also in Asia as well. So that's calling one thing out, and this is where we need to get better as a whole group to advocate this type of need. So I talked about male female. Another huge issue, I believe, and is also reflected very much in when we're collecting samples for generating iPSCs is also an issue for us, is that so this is the Alzheimer's clinical trial, and this is lecanemab, which actually has been approved for use in Alzheimer's disease. I won't go into the it's quite a political hot potato, that one, and it's not really my area of expertise, but it has been approved. But what was pointed out, and I think is really interesting, is that the major participants in this clinical trial were white, and a small population of that clinical trial were Asian, and an even smaller population were black. And we know there is, and from quite a lot of literature in the past and quite a lot of work that's been done in the past, that there is differences between race and ethnicity, and effects of different diseases affects different races more because of genetic predisposition to certain diseases. So again, I'm very much calling out that if we want to do clinical trials in a dish, we need to, we're certainly aware, and we're trying to ask for more participants from different backgrounds and different races and ethnicities to come forward to participate in generation of iPSCs, which is what we work, but I would also advocate in the clinical trial space as well, because we're not just trying to we're trying to identify drugs that work for everybody, not just for specific individuals. So that's drug discovery. I've just got a couple of slides on, or one slide, I should say, just on iPSC cell therapy. It would be remiss of me not to point it out. So drug discovery is always moving forward, and iPSCs are being used and are being looked at for use in cell therapy, and have been used and been put into patients already. And I'm just going to highlight this one slide. So iPSCs have been used and are being used for Parkinson's disease, and what we're talking about here is people putting neurons into patients with Parkinson's disease and seeing if they engraft and help. And the one underneath is very much ongoing work, and has been ongoing for quite a long time, in terms of, can people add iPSCs to hearts to enable engraftment and try and treat heart failure. I think both of these are ongoing work. The picture on the right-hand side highlights a number of companies that are working or trying to work in this space. I would say there's still quite a long way to go. There's probably a number of bumps in the road to get to a point where cell therapies are able to be used. But obviously the value and the opportunity is not missed, and certainly by all these companies and pushing forward in that space. So just my last slide. So Stephen is a somebody who is working with us at Axel. He's part. He works in Edinburgh University. He has Parkinson's disease. He does very much push forward a whole process around using stem cells, and is very, very interested in the work that we do, and can see stem cells adding value to people with Parkinson's. He's also acutely aware that the likelihood of us and other companies identifying compounds that is going to help his Parkinson's in the short term is pretty low. But he's very much pushing forward and saying, please be quick. I can see the opportunity the iPSCs add, and please use this technology to develop better drugs for patients. Thank you very much for listening to my talk today, and I'm back to Vanessa.
Thank you, Ashley. This is very mind-opening for me. I mean, my background was a little bit on this field. You know, we start from thought leadership, as you famously know, and then we move to the Yamanaka factors and to reach to where we are today. I think this is fascinating how the field has progressed, and there are still many things to do. That's what I hear from your talk. We do have a few questions on the Q&A panel. Maybe I will just start, with a bigger question first: is how scalable is iPSCs? And is this actually a feasible model as an alternative to animal models?
So theoretically, infinitely scalable. So in terms of iPSCs themselves, generating vast quantities of those cells is possible. I would caution that a bit with regards to the QC. So the QC is really important. That's why I highlighted it. But certainly from the work we're doing and work that is published and work that we see at conferences, being able to do what we talked about there, clinical trial in a dish, is certainly not as far away as one would think. Sample collection is one. There are some recent papers out there from Mount Sinai Hospital where they have collected, and patient advocacy groups have been really important in this space. They've collected 1,000 ALS samples, and they've generated iPSCs from those and generated motor neurons from those. And they can trace back the clinical information associated with those. So if you think about the scalability, it certainly is possible, and it's certainly being done at the moment. So is it scalable? Yes. Are there challenges? Yes, there are. But is it being done as we speak? It absolutely is.
So this is a very nice segue to the question that Vishal has on the Q&A. Is there any in vitro methods or assays that has used iPSC as a validated and accepted method for FDA to replace animal studies?
As it stands right now, I'm not aware of any where somebody has gone and replaced animal cells, still replaced animal cells. I think the one space where they are being used on a far more routine basis is the cardiomyocyte space, and have been used on a routine basis in the cardiomyocyte space. I think that's well accepted technology. It's probably the most veteran, should we call it, iPSC technology that's been around. And because of that, there's a far more acceptance of it and its usage. Is there an actual example I can think of right now today? No. Part of that is driven by very much driven by the regulators. So the regulators will say, and have said up until I talked about the FDA point there, have always said you need to be able to prove that your compound is safe, and rightly so. And the way that we say you need to do that is animal models. And until And so the use of animal models or animals in drug discovery is always going to be driven by the requirement from the regulators. And until the regulators have turned around, like the FDA, and said, actually, you don't have to use animal models. They're not saying you do not use animal models, but what they're saying is, or animals to do your modeling, you don't have to. I think one of the areas that I think where that's coming from is, I believe, is coming from the cell therapy space. I think it's really difficult, if you at a high level, to understand if you're implanting human cells into a patient with Parkinson's disease or a patient with cardiac failure. How do you even think you can model that in an animal model, or that the relevance of the animal model has any value? I can't see how that makes any sense at all. So I suspect there's certainly a drive from the cell therapy space, which is far more complex and very different than putting a small molecule under your tongue, driving that. But both will drive to better models yet. So modeling cell therapy, the sort of cell therapy I talked about, not talking about CAR-T and autologous cell therapy, but the sort of cell therapy that I talked about, I think is almost impossible to do. So why do it?
Fantastic. You know, it's interesting how you talk about women. It's not well represented in many of the clinical trials, and today happens to be an International Women's Day. So my question for you is that in the lack and the understanding of this, you know, some kind of iPS cells bank that is needed to then be representative, is there a global initiative or global effort to drive an iPS cells bank like what the Japanese are doing?
So there are banks that are available. There is certainly banks that are available, as you say, in Japan. There are banks that are available in Europe too, that can be used for drug discovery. I think in terms of it can get quite complex. So the banks that are available for individual groups to use at a research level are fine. If you then try and use those in the commercial setting, there can be quite some legal issues associated with some of those. And some of that is also driven by the ethics. So I didn't talk about ethics at all, and sample collection. Sample collection is a really complex area too. So I think when you look at some of the original ethical approvals that were used, things such as CRISPR editing did not exist. So because that came around in 2013, right? So the way the ethics were written in 2006, 2007, 2008 in sample collection, because people were still using primary cells in those times, did not recognize gene editing, or whole genome sequencing in some cases is not recognized either. And so you try and use a sample, or you have to be very clear about when you do your sample collection, or you use an old fibroblast, whether you have the opportunity to use those samples. So ethics is one thing. We do know, so we are currently collecting samples and generating new iPSC lines as part of the work we do for certain clients. And we do know of where patients want. So once we write the ethics form and make it very clear, we do know of patients that are just not happy to have their genome sequenced and used for that type of purpose. So not only is there banks that need to be generated, it's if banks are generated, are the ethics correct to allow you to use those samples in the way that you want to use them, and understanding that. So that's another area which is again, I haven't talked about at all, but you know, is an area of importance.
Okay, I'm just mindful of time. We are at 6:01 now. Do we want to just take one last question?
Take one last question.
Okay, I'll pick a hard one. So Just a quick one. Can you comment on other drug testing models, like your in vitro 3D organoids versus your humanized mouse model? How do you think these different models, together with mouse models, right, can come in synergies to create a better representation of a human disease?
So I think one of the things that we have, what I talked about co-culture models and putting those models together. So if you look at what people have always done until fairly recently, they've always gone, so Alzheimer's disease, let's say, associated with neurons, neuronal loss. I need to look at neurons, right? So the thought process is, I'm losing my neurons, I need to look at neurons, but actually it's probably not neurons that are driving it. Neurons are the outcome of the disease, whereas microglia and astrocytes and oligodendrocytes are probably really, really important in that disease pathology, and certainly neuroinflammation as an area for study for Alzheimer's disease is growing, and is certainly an area that people are focused on because they think that's probably where the issues are. So co-culture models in whatever form are absolutely going to be really important to generate better models associated with better outcomes and better outcomes for patients, i.e. better drugs. Now, whether that's from 3D scaffold-based co-culture models, some of which we carry out, whether that's from organoid-based models, and depending on what disease you work in, spheroid-based models. So cancer has very much pioneered, I would say, spheroid and organoid work because of the activity of compounds or lack of activity of compounds in 2D and the difference between 2D models and 3D models in cancer. Some of that translates into other diseases, sometimes it doesn't. But I would say there is never going to be one answer that fits all. They all add value to a lesser or greater extent. And so is a spheroid or a organoid model better or worse than some of the models I showed there, some of the models we're working out? No, they will probably all complement each other.
Thank you very much. Thank you, Ashley. I'm so sorry we couldn't take all the questions that's been listed, but we will funnel them to Ashley. And if you have any questions, burning desire that you want to find a little bit about Excel, you can always write to them. Last but not least, we do really like you to take out your mobile phones if you have one, or to scan the QR code that you see on the screen and give us some time to take the feedback. What do you think about this event and how can we do better? And what are the topics that will interest the wider audience here? If not, Ashley and Duncan, thank you very much for Excel's participation in this learning series for drug discovery. I think the ecosystem has learned a lot from you. I mean, I love iPS cells. I was a PhD student when we first heard about Yamanaka factor. So it's mind-blowing to see how this whole landscape has continued to evolve in exciting phases. I think that there are many impetus, especially from the FDA side, pushing the 2.0 since November 2022. I think there's much, much more to be seen how we can use different disease model to do drug development more efficiently.
Absolutely.
Thank you, everyone. If there's nothing, we will close the session. Thank you, Ashley. Thank you, Duncan. And thank you, everyone, for staying with us today.